Hello friends, welcome back to NitVed Bioscience. This is a quick revision video on the Ramachandran plot. We know the primary structure of the protein. It is a string of amino acid where all amino acid are joined to each other by the peptide bond. To make the secondary structure from the primary structure, this primary structure has to undergo the folding. Common example are like alpha helix, beta pleated sheet. So here we can see that after the folding of the primary structures, we will get the secondary structures. So this primary structure has to undergo the folding, means bending takes place. So there are the three types of the bond present in the primary structures. The one between the C and N, that is known as a peptide bond. Apart from the peptide bond, there are the two other bonds are present, that is between the C alpha and N and the C alpha and C. But this bending cannot take place at the peptide bond because there is a partial double bond character present at the peptide bond. Therefore this peptide bond is rigid, therefore it is not involved in the bending. Bending takes place at the other bond apart from the peptide bond, other two bond, that is between the C alpha and N and the C alpha and the C. Okay, so after the bending, the angle formed between the C alpha and N is known as a phi, and the C alpha and the C is known as a psi. Together it is known as a dihedral angle. Okay, how to remember this? So C alpha and C, so C, psi. If we remember the one thing, so we can remember the other. So between the C alpha and N is phi and the C alpha and C is a psi. So the degree of the rotation is the range is minus 180 degree to plus 180 degree. This minus and the plus sign is for the clockwise and the anti-clockwise rotation. But thus all the possible angles between the minus 180 degree to plus 180 degree are present in the secondary structure? So the answer is no. Why? Because we know the primary structure, the structure of the amino acid. In the structure of the amino acid, the side chain is present. Okay, so due to the steering hindrance of the side chain, due to the bulkiness of the side chain, if the side chain is a more bulkier, so it will have the more steering hindrance. If the side chain is a less bulkier, then it will have the less steering hindrance. In a simple or the common words, we can say that if it is a more crowded, so there will be the more restrictions, and if it is a less crowded, then there will be the less restrictions. So this considering this fact, there is a Ramachandran plot. So this Ramachandran plot, it helps to visualize the allowed conformation because of the steering hindrance, not all the possible angles from the minus 180 to plus 180 degrees are allowed. Okay, and so which are the allowed regions, not allowed regions, so that we will see, and the some regions are the most allowed regions. So all these things we can see on the Ramachandran plot. Okay, so this is Ramachandran plot. On the Ramachandran plot, on the x-axis there will be the value of phi, on the y-axis there will be the value of psi. Psi resembles the y, therefore we can remember it. Okay, so the range is from the minus 180 degree to plus 180 degree, and here from the plus 180 degree to minus 180 degree. These are the allowed regions. Okay, inner ones, these are the most allowed region. The upper boundaries are the less allowed regions, and the remaining, it is the disallowed regions. The angle, so the upper left quadrant has a allowed region for the beta sheet, upper left. Upper right has the left-handed alpha helix, lower left has a right-handed alpha helix regions, means it is a allowed region for the right-handed. So upper left beta sheet, upper right left-handed alpha helix, lower left right-handed alpha helix. The first quadrant will have the value in the minus plus, second in the plus plus, third quadrant will have the value in the minus minus, and the fourth quadrant will have the value in the plus minus. There are the two special cases. One is a glycine and the prolines. So in the side chain of the glycine, hydrogen is present, so it will have the less restricted regions and it occupies the more region on the Ramachandran plot, while in the proline the ring structure is present. Therefore because of the presence of the ring structures, it is a highly bulkier amino acid, therefore it has a very restricted regions. And what is the importance of the Ramachandran plot? Mainly it is helpful powerful tool in the secondary structure analysis and plays a crucial role in understanding the protein folding, structural biology and the drug design. Thank you.